Good afternoon, everyone, and welcome. My name is Cheryl Reynolds with the UC statewide IPM program, and welcome to the UC Ag Expert Talk on citrus dry root rot. Peter Casina is also here with me, and he'll be running the polls and troubleshooting any questions or technical problems that might come up. And please note that the webinar is targeted to growers and pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, are not to be followed without a clear understanding of their legal use by homeowners. Okay, and so now I'd like to introduce our speaker today. Dr. Akif Eskelin is a plant pathologist and cooperative extension specialist at UC Davis, and today he is speaking on citrus dry root rot. So now, Akif, I'm going to pass this over to you, and you can go ahead and share your slides.
Okay, so good afternoon, everybody. First of all, thank you very much for joining us this afternoon, taking time from your busy schedule and listening to our webinar session. So I'm pleased to give this presentation on dry root rot disease of citrus, which I have done quite number of years works on this topic and subject when I was working on citrus diseases in my previous position at University of California, Riverside. So I would like to start with the name dry root rot before I go through to all the details and everything. So this dry root rot disease, first of all, it has given a very good name, dry root rot. And in the plant pathology discipline, when we are naming a new disease, we try to give a name that could describe the symptom, how the disease look like, so that will give an idea to the growers or peoples to what to look for, how to identify the diseases. The reason that I'm saying that the dry root rot is a good name is that when you see a rot on a wood, usually it's the soft one, which is very common. The concept of the disease is the soft rot. So in this case, which caused by a complex of the disease, it's a dry one. So it's like not the canker, which is that tissue, but it's the dry root rot. So with that, so I would like to start give my presentation on this subject. First of all, I would like to thank to my co-authors on this the subject that I have worked with, Tony Adesamoya, who was my postdoctoral scholar when I was at UC Riverside. So he has now another position in another part of the United States. I also would like to thank the Ben Faber, Gary Bender, Neil O'Connell, and Craig Carlson from University of California Cooperative Extension. So with their help, we were able to get some more information about the causal agent of the dry root rot, biology of the pathogen, and then management strategies of the disease complex that our citrus growers have been dealing for a long time. So another point that I would like to point out is that the dry root rot disease is not a new concept or not a new disease of citrus. It has been known for a long time. Before myself, Dr. John Mange has done a lot of work, who was a professor retired right now from UC Riverside. Later on, his graduate student, Gary Bender, has also worked on this problem for a while. So Gary is also retired emeritus at this moment. So with that, I would like to go ahead and then start telling you the story behind this dry root rot. So it's a very common disease, which I also call it disease complex of the citrus. So in this slide, as you can see, the one of the tree completely died. So later on, I'm also going to talk about quick decline of the citrus, which is similar concept, similar causal agent, but it's a little bit more complex than the dry root rot. So when the dry root rot symptom, as you can see, completely trees is completely died. Most cases, the tree dies within a short time. For example, in the plant pathology, when I'm identifying the diseases, when I look at the symptom on a dead plant, as you can see on this slide, the plant leaves are still intact on the plant, but completely plant is completely died, which tells me that this plant died within a short time. It didn't have a time to defoliate their leaves. So that's the quick death. So that is one indication to me that it's something going on. Some pathogens act very quickly and then kill this plant within a short time. Not only pathogens, probably some of the environmental conditions also could affect and then kill this plant within a short time. In this case, dry root rot is the one. So anyway, so another indication that tells me that the dry root rot is the soil-borne pathogen or group of the pathogen that cause. In this case, that's the case, that's dry root rot. When I see entire plant shows the symptom like this, that also shows that there is something wrong in the either main trunk or in the roots of the plant. So dry root rot is not the disease that causes only one single cultivar. So it also can show up on more than different cultivar as well. So in this case, you can see the two trees. The one on the left starts showing the symptoms. The one on the right is healthy looking. So within a short time, the one on the left with the high temperature during the middle of the summer could die within a short time, quick time. So my point here is that it is very difficult to predict or see the sign of dying plant from the dry root rot in the field sometime. I'm sure if you are a grower, if you have this problem in your the citrus orchard, I'm sure you have experienced it. So the tree one day looks so healthy, no problem, and next day they can start showing the very weird dying symptoms as well.
Akif, did you want to bring up that first poll?
Yes, this is the test if you are listening to me or not. There you go. So first one is dry root rot a new disease of citrus? Yes or no? Very good. Yeah, it is not a new disease of citrus. It has been known for a long time. It is caused by a complex of the pathogen plus environmental condition that I'm going to talk about it during my presentation. Very good. So, as I mentioned, that this is not only one cultivar or variety of the disease. It could cause on lemon. It depends on where you are. It could also cause on the navel orange. We have seen on the sometime in the mandarin, but it's not that common on the mandarin. The most lemon and navel orange that we have seen. So I'm glad that there are a lot of the people are attending from the Southern California, which the climatic conditions could be a little bit different than the coastal area, where you can see these kind of symptoms more often than in other places. So our second question is, which variety is more susceptible to dry root rot? Questions are, the alternatives are, all the citrus varieties, only lemon, or only orange, navel orange. Okay, yes, that's true. All the citrus varieties are susceptible. You are also right if you say lemon and orange. Yes, both of them are susceptible as well. So, but all the citrus varieties, yes. Thank you. So these are another symptom of the dry root rot. The one on the left is the start dying. You can also see some greenish leaves on that one. The one on the right in the rows is completely dead. So sporadically you could see this kind of symptom in your citrus orchard. I just wanted to give you the different scenarios of the symptoms in the field. So basically when you see that plant, it's most likely caused by the dry root rot, which is the quick decline of the citrus. So what is the causal agent of the dry root rot? So in the research that we have done is that or previous research also confirmed that the Fusarium solani is the most commonly isolated fungus from the disease wood. So this is the one of the tree that you can see on the soil line. So sometimes without making this kind of cut or peeling, when you look at the soil level, you may not even notice the dry root rot symptom. But what you can also notice is that some sunken areas at the soil level. That is also indication that there might be something going on. The reason that you have that sunken point in the bark is that when the tissue dies by the pathogen, when the rest of the tissues are still alive, the living tissues continue growing while the dead tissue stay as it is, and then it shows a sunken point. So, so without peeling it, you may not be able to see the discolored area. So when you see that kind of sunken area with a clean knife, just peel the bark as it is shown in this slides, that you will see the dark symptom discolored area. So which is the indication of the dry root rot at the soil level. So to me, so only one side of this tree is showing the symptom. So there are some other part, the living. So probably this plant is looking okay for now, but it can collapse anytime during the middle of the summer. So this is another scenario. It's a little bit worse case than the one on the left. The one on the left is look like the 50%. The one on the right is completely rot. And so actually you can also see some of the brownish color. This is very important. When you pull this kind of plants, if the color black or purple, which specific the purple, Fusarium solani, when they colonize in the root, they show kind of like the purplish kind of the symptom in the plant. So another indication of the dry root rot is that when you look at under the soil level of the trunk, you see the more wet spot, wet soil, which means the root of the plant is completely dead. The plant cannot take any moisture or water with their roots. All the irrigation water that you give and on the soil level stays there, make the wet point, which also encourage or stimulate the dry root rot advance within the plant. That's also an indication that you can have an idea of what's going on in the soil level. So let's bring up our third question. So what is the primary cause of dry root rot? Is it Fusarium? Is it stress, including over-irrigation, mechanical damage, or other diseases, or Colletotrichum? Yes, Fusarium is the primary cause, but other stress factors, for example, that answered the question of Ben, over-irrigation. When you have the over-irrigation in the plant roots, as other living organisms, they require oxygen. They need oxygen. So that means the soil level has to be aired out with the irrigation. So when you do the over-irrigation, plant cannot to get the oxygen. So that will also stimulate some of the pathogen to advance within the soil level and then cause or speed up the dry root rot process within the plant. The second one is the mechanical damage. Mechanical damage that could cause by the cultivation equipment, that could cause by some other rodent animals, that could open an entry point for the pathogen to get in the plant and then advance it out. Let me put it another way. Fusarium Fusarium solani is very common fungus, soil-borne fungus. It's everywhere, but they are not aggressive pathogen. They are opportunistic pathogens, which means when the right conditions comes in the plant with the environment, they could be aggressive pathogen. What are those right conditions? When you make the over-irrigation, when you have the mechanical damage in the soil level, when you have the other diseases stress, those are the ones that encourage. But Colletotrichum is not the causal agent or part of the disease complex. It is another group of the pathogen that cause other diseases on the shoots and then canopy level of the plant. It is not associated with this dry root rot. Okay, all right. So as we said that the Fusarium species has been in all over in the soil, contaminated. They are present. They are just waiting for right conditions to come that they can colonize the plants. So which means they are soil-borne. They can stay in the soil for a long time. They figured out during the evolution how to survive in the soil. So most of the soil-borne pathogens produce some kind of the overwintering structures within the soil, whatever their environment is, so they can stay for a long time. So in this case, Fusarium may stay on the dead wood, that part of the plant. They can even stay as a conidia in the soil mix. Again, so the one on the left that I'm showing here is that it shows the very good profile symptom of the dry root rot. As you can see, just at the soil level, the lower level is completely dry, dying, and then above the soil level looks good. So sometimes when you look at and then find that this kind of discoloration on the soil level, and when you look at the plant, is showing some green shoots and symptoms, you would be surprised how this plant is still alive. You know what's happening? This is what's happening. So when the Fusarium start colonizing from outside inside, so some of the xylem roots, part of the plant is stay alive in the center. They still continue pumping some nutrient and water to the leaves. So for a while, plant leaves get what they need, even though it's limited, and they continue photosynthesis. All the photosynthesis products supposed to go to either fruit, to the roots, through the phloem, and then make the new feeder roots, right? So when the Fusarium dieback girdle from the outside to the inside, the plant cannot send any starch into the root, and then they cannot produce the feeder roots. That's why they could stay green for a while their the root crown is showing this kind of symptoms. So right environmental factors comes, which is during the summer in California climate, they could just finish it up and then show the symptoms. So dry root rot is not the problem for the mature plants, but it also could show on the younger plants. In here, the one on the right that you can see a little bit younger plants is also showing a lot of the dry root rot symptoms. So with that, let's bring up our question 4. Is dry root rot a soil-borne pathogen? Yes or no?
And there is another question for you from Matt. And he asks, is the purple discoloration seen for rot associated with the stress response or anthocyanins from the plant?
That's a very good question. I don't know. But what we haven't done any kind of analysis that one. In my opinion, I have seen that one with the other Fusarium species. I think when the Fusarium colonizes in the wood, that's the secondary the metabolite that they can release into the root. We have seen similar color when we are culturing on the some of the Fusarium solani species on certain media. They also release that kind of symptom. So saying that, so that's just observation. We haven't done any the analysis whether it is the response from the plant or the fungal itself is producing some secondary metabolite. In my opinion, that's the secondary metabolite from the fungus. Yes, dry root is a soil-borne pathogen, and then that's the answer. Okay, let's move on. So as I mentioned before, that so this is the internal symptoms. This is the cross section of the one of the citrus tree that has the dry root rot. As you can see, like almost 60 or 65% of the plant completely died. That's not functioning. Only 30% seems to be alive and functioning for a while. Again, this citrus, this kind of condition, could stay alive and productive kind of for a while until the right condition comes that just give up on that one. So this is another good example when you look at the cross section or longitudinal section of it. So the one in the lower level is the rootstock. As you can see, the Fusarium moved through the rootstock, and then this is the graft union, and then can also move up the graft union and then get into the sign. So it doesn't matter whether it goes to the sign or not. So it has already done its own damage on the rootstock level anyway. So that plant would die. So these kind of discoloration, blackish or the purplish, is the very characteristic. So these are not like the common symptom for the Phytophthora, because Phytophthora usually show up and cause damage on the feeder roots, and then later on other secondary pathogen can get in and then colonize the main root as well. So these are the one that could give you an idea what kind of problem you may have on your citrus. Okay, with that, let's bring up the question 5. Can you identify dry root rot symptoms in the field easily? Yes, yeah, you can have an idea about the dry root rot just looking at the canopy and just going into the soil level and then looking with your sharp knife on the soil level to see if you are going to see that discolored area. That will give you an idea. But sometimes when the symptoms are not obvious on the soil or ground level, you may need to take some samples to the laboratories to confirm it as well. So that's what I will say. If you are not familiar with the things, when you see your plant died quickly, I will always take the samples, go to the plant pathology laboratory, and then have them confirmed whether you are dealing with the dry root rot, which is known, or you are dealing with something more or different than the dry root rot. It's always good to know what's going on that part of the plant. So earlier, as Ben also asked the question, like I answered some of those questions. So this is the cross section of the wood tissue that was infested by the Fusarium. I just want to point out that you all know that the wood part of the plant has xylem vessels, where plant takes the nutrient and water, pump up to the upper level of the plant when they make the photosynthesis. When the plant is infested by a pathogen, it could be fungal, it could be bacteria. What they do is that they respond to that infestation. Their goal is to prevent the movement of the pathogen, whatever it is. In order to do that, plant try to produce tylosis and gumming in their xylem vessels, that in their mind, they are trying to, in their defense mechanism, they are trying to prevent the movement of the pathogen. As you can see in this picture, there are some xylem vessels open, there are some xylem vessels brown, it's closed. That's the vessels that was produced by the plant, and then they just plugged it. Actually, that over response is the one that caused the dieback or declining on the plant. If you could tell plant to be relaxed, it's gonna be okay, maybe they are gonna be okay, but it's not gonna work that way. So which means sometimes plant are overreacting, like how we have the kind of overreaction allergies, that the kind of like that one. Plus, if the pathogen is most of fungal pathogens, they can also colonize within the xylem vessels to move up to the plant, and then they can also pluck mechanically in that the xylem vessels. So in this cross section of the wood tissue, I'm just trying to show you that so some tylosis and gumming could be produced by the plant, and then to prevent the movement of the pathogen, that is the one that could cause the dieback on the plant. So this is the longitudinal section of the xylem vessels. Actually, you can see that the xylem vessels are, I just want to explain that part. It's not the continuous kind of hose. So the xylem vessels is made by the pit membranes. Pit membranes is the membranes that the plant produce to prevent the movement of the pathogen, actually. So it's not the continuous endless the hose, as you can see here. So in that area also plant could produce some gumming and tylosis to prevent. So these brownish the colors are showing the response from the plant. Anyway, point here is that so these are the internal symptoms. So it is like complex of the disease, not only caused by the pathogen, but also other environmental conditions, including tree response to the pathogen invasion. So when we first wanted to get more information about this dry root rot, which has been always seen around it, and then we also had the access to, excuse me. We also had access to some molecular identification techniques in the university. We wanted to make sure to identify the primary causal agent, to make sure if we are dealing with the one Fusarium or more than one Fusarium, which I'm going to talk about. So we have done a field sampling, or I will say field survey by the funding that we received from Citrus Research Board at that time. So we did the survey on 6 different counties, 22 different locations. From that field sampling, we collected more than 2,000 samples. We made isolations from each samples that we collected at that time. We ended up having more than 4,000 cultures, and then we identified them based on their morphology, which we store them in the long-term storage, and then we identified them based on their either morphology or also molecular identification techniques. So at the end of our survey, we ended up finding more than one Fusarium species are involved with this problem. Fusarium solani is the primary one. We also recovered Fusarium oxysporum. We also identified Fusarium proliferatum, which is very well known aggressive pathogen right now. There are a lot of reports coming out from the proliferatum, and also Phytophthora. Phytophthora is also a contributing factor to the dry root rot. As I mentioned earlier, that the Phytophthora usually attack and colonize the feeder roots, which is the main roots for the plant to get the nutrient and water. When the Phytophthora knock it down, and then plants start having stress, and then the Fusarium friend is staying in the soil waiting for the stress factors to come up, and then they just colonize in the plant. So which means that not only one Fusarium species, also Phytophthora is involved in this problem. The one on the right bottom corner, you can see the conidia, which is the spores of the Fusarium that you can see on that one. So with this, let's bring up to our question 6. Is Phytophthora root rot involved with the dry root rot, the problem? Yes or no? Yes. As we said before, so Fusarium is not only one contributor. There are other pathogens contributing this process. Okay, let's move on. As I mentioned that we were curious about how many Fusarium species were involved with this problem. I had to mention that Fusarium species are also known as complex. It is also known as the Fusarium solani complex. With this sampling, we identify a group of the pathogen from the citrus growing areas in California under the Fusarium solani name. We also isolated other group of the Fusarium, which is also known as Fusarium solani, but in this phylogenetic clade, it is separating out a new Fusarium species. At that point, I'm not a taxonomist of the mycology, so we didn't do further identification or naming of this Fusarium. It's not my expertise area, but we pointed out that must be another new species that needs to be studied on this Fusarium solani complex. So with this, the phylogenetic tree, I didn't mention that. This is the family tree of the Fusarium species that we isolated from our survey, which is kind of like based on their DNA, which is the true identification. So this shows how each species or each fungal Fusarium species are related to each other. So this is showing that there are two clades separating from each other, which shows that this could be different than the Fusarium solani. So we might have more than one Fusarium solani in that. Is this Fusarium new? We don't know. Maybe this has been in the valley for a long time that we were just able to identify because we have now access to molecular identification techniques. So for the molecular identification, we use the ITS with the phylogenetic tree, and now we are not satisfied using the one single loci in the DNA, so we are able to utilize more than one loci, combine, and then get the fingerprint identification of it. That's what we did. This is the quick results frequency of the recovered pathogens from our survey. As you can see, so this is the number of the organism that was recovered. We have isolated bacteria, group of the Deuteriella from our survey. Fusarium solana is the primary one. Other Fusarium species, I just talked to you about it. Phytophthora was involved, sorry. Phytium and other soil-borne pathogens, and we got a lot of saprophytes. So this is the results is showing that even though Fusarium solana is the primary pathogen, but also other pathogens could be contributing this dry root rot problem in California. So, co-postulates. In order to claim a fungal isolates to be pathogen, you have to complete co-postulates. It's the name of the guy who first came with this idea. So what it means is that whatever you isolate from the plant symptomatic tissues, you have to put them back into the healthy plant to see if they are going to show the same symptoms. Not only with that, you have to recover the same pathogen or same fungus that you put on that healthy tissue to complete that cycle, that whatever you isolated, that's causing the same symptom, and it is there when you recover it. So it's called that process co-postulates. We wanted to produce the co-postulates in the healthy plant with the Fusarium isolates that we isolated. So what we did is that we found like the healthy looking plants, we inoculated them, and then these are the results from the pathogenicity test. Fusarium solani that we isolated, that was the most aggressive one. Other Fusarium species also showed some the degree of the aggressiveness comparing with the control. So that also showed that the Fusarium species that we recovered from the field survey showed that they are pathogenic to the citrus with this trial. So I also would like to talk to you about the citrus quick decline. As earlier I mentioned that this is also the part of dry root rot complex. So a few years ago, during one of the hot summer in Central Valley, in Tulare area, we got a call from the farmers that their citrus trees are dying within a few days, within a couple days, as you can see from the one on the left. And so they wanted to identify the causal agent of that. So me and another extension specialist, Dr. Georgios Vidalakis, he's a virologist, we did a quick survey in that quick decline showing areas in Tulare area to identify what is the primary cause of this problem at that time. I just want to point out that one. So in that quick decline survey, what we have noticed is that some of the declining trees are showing some abnormal the Setici symptoms at the graft union on the bark and also on the wood tissues, as you can see the picture on the left. The one on the right, healthy looking plants, you don't see that symptoms. So these kind of symptoms are very well known that could initiated by the viral diseases. So that's why Georgios, as a virologist, and I, mycologist, wanted to get together and then address what is the primary cause. We also had some ideas what is it causing, that is also known concept, but we just wanted to prove what it is in decline. Okay, so as you can see, those are the symptoms. Okay. So what we did at that time is that we visited 5 different orchards and collected samples from the declining and healthy looking plants. So Georgios did some citrus tristeza virus, the identification. I did the Fusarium solani or mycetes as the mycology, and also we did the iodine starch test. Iodine starch test is very well known test. It is the iodine that's, as you know, that's the same iodine that you know. When you put the iodine on the wood pieces, it just reacts with the starch within the tissue. I'm sure in your high school you have seen that kind of test on the potato starch test. This is the same test that would give you an idea whether that tissue is starch depleted or not. That's why we wanted to get an idea. So why we wanted to get a starch depleting test, I'm going to talk to you about it. So from those samples, this is the starch test that you can see up. The one on the left, there is no oxidation, there is no discoloration, that means negative. The one on the right has starch, okay, so positive, okay. From the samples at that time that we collected, that all the declining trees had same results: CTV positive, solani positive, and lack of the starch. Orchard 1, orchard 2, orchard 3, and orchard 4 is all showing the same declining. Okay. When we look at the starch test, they all show the lack of the starch. The reason that we wanted to look at the starch test is that the Fusarium solani is known to attack and colonize starch depleted roots. That's what they like. In that one. So that's the one. So when we look at the declining trees, they have the all same pattern, combination of the CTV and Fusarium solani. Sometime in some healthy looking plants, we also found Fusarium solani alone, but no declining, healthy looking. Some cases in healthy looking, we only found the citrus tristeza virus positive, but no solani, but plants was looking healthy. So which shows that combination of the CTV and also solani is the one that's showing the decline. So what's happening is that during the winter, the citrus roots are high in starch. During the summer, high in sugar. Plus, citrus tristeza virus is a phloem limited virus, and when it colonizes on the effect on the bark or phloem, that prevents plant to move the starch into the roots and then make the roots starch depleted. Therefore, it makes the roots susceptible to the Fusarium solani that has been waiting for opportunity to get into the plants. That's what is happening. So that combination of the right condition with the right pathogen within the soil that caused the citrus quick decline. So if I had to summarize it, so dry root rot is a disease complex. Citrus quick decline is also disease complex caused by the combined effect of the Fusarium and then citrus tristeza virus. I have to point out that the sour orange rootstock is the most susceptible to the citrus tristeza virus. But I believe farmers still like to use some of the sour orange because it's good for phytophthora, it's good for the tree response, it's good vigor very good rootstock, but it's very susceptible to citrus tree virus. Still, I believe some of the growers are still taking the risk using some of the rootstock as well. So I believe there's a citrus tree the committee established by the Citrus Research Board. They are doing the outreach and education to warn farmers that stay away from that rootstock, which is very susceptible to the citrus tree virus. It's a very interesting concept. So sometimes if you don't see that problem, you can also see that could be the source of the inoculum for the other farmers next to you who hasn't been using the sour orange. It's very different, interesting concept. So our growers should follow the guidance that is provided by the scientists and also Citrus Research Board to apply that. So once in a year, once every 10 years, 20 years, with the high the heat in the middle of the summer, we have seen these problems in Tulare. So with that, let's bring out our question 8 to see what it is. Okay, so what's the cause of citrus quick decline? We just talked about it. Is it Fusarium itself? Is it citrus tristeza virus itself? Or combined citrus tristeza and Fusarium species? Okay, that's correct. It is combined effect of the citrus tristeza virus and Fusarium species. So that will give an idea what to do, how to do. So all these results or part of these results that I talk was published in Sutra Graph. If you want to read more about it, please feel free to go. You can also find the copy of these articles in my lab website in Escalon Lab at UCANR website. So with that, I would like to talk to you about the management. The reason that I'm saying the disease management, there is no the single bullet, silver bullet kind of the control method. There is no pesticide or fungicide application that you can apply and then be done with it. It is the management. So which means irrigation should be done with care, because we now know that the too much water, too much irrigation could initiate the initial the dry root rot process. Okay, drainage is very important. It is the same concept. When you have the standing water around the root area, that will also weaken the plant, get rid of weaken their defense mechanism. Also, they can also activate the Fusarium solani within the soil level. So movement of the equipment is the one that could also spread the disease, because we now know that the Fusarium solani is soil-borne. The equipment could also cause mechanical damage and then could spread the disease from one location to another location. You have to be very, very careful about it. So proper fertilization could minimize the disease. The reason that the stress factor is very, very important. Even if you have like the initial infestation, when you take care of your plant very well, you may extend its lifespan a little bit more, that you can get a little bit more the yield while you are doing it. So as I mentioned before, rodent control is very important. Herbicide use, we have a lot of seen that some drifts from herbicides that affect the other plants, that cause a lot of stress, and also other chemicals could also cause some stress in the plant. So it's very, very important. Sanitation is very important. When the plant is died or dead, you should remove them and then clean that area to prevent the further infestation. So last but not least, Phytophthora should be the part of the management strategies. It's very important. In California, we have two kinds of soil-borne Phytophthora species. It's very common. So the management of the Phytophthora will help dry root rot management. So with that, let's bring up our question number 9, which is the last question. What fungicides can you apply to control dry root rot? DMI group of the fungicide, or soil fumigation, or no pesticide, cultural practice zone? Okay, that's correct. So there is no pesticide application directly. So you can control some of the Phytophthora, but there is no, like the pesticide that you can apply to control the dry root rot. So it is a management practices starting from the well taking care of the plant, not over irrigating, and then all the disease management that I mentioned will help keeping your plants. And then stay away from the sour orange rootstock, even though it has a lot of benefits, but it is very susceptible to citrus tristeza virus. So I will stay away from that one as well. So with that, this is my acknowledgements. So during my time at UC Riverside, so Citrus Research Board funded almost most of my research, and then we have we have, I would say we have demystified a lot of things that we were curious about it. So I would like to thank to the Citrus Research Board, as well as the citrus California citrus growers who opened their door and then farm for us to do all kinds of survey, experiment, trial. So I miss them a lot in my new position. So I would also like to thank to all the farm advisors that I have worked with: Gary Bender, Mary Bianchi, Ben Faber, Craig Carlson, Neil O'Connell. I also thank to pest control advisor Joe Barcinas and all my lab crew. So with that, if you have other questions, I will be happy to answer your questions.
Okay, I do have a comment in chat from Dustin. He says, I see some companies stating that they have beneficial bacteria that will fight this. Any experience with that?
I don't have any experience with any beneficial bacteria, but I have an idea about the beneficial bacteria. Yes, so my lab was also doing some studies on other crop on beneficial bacteria. If the question or companies are talking about the endophytic beneficial bacteria, which always help plant induce their defense mechanism and then fight against the pathogen better. If in that concept, it may have an effect on it. Being said that, I have no trial, I have no experience about it. I'm just sharing my opinion on that. So yes, beneficial bacteria is as a common sense. It may increase the defense mechanism of the plant against any kind of pathogen, not only dry root rot, but that has to be studied.
Okay, so I don't think we have any other questions. So thank you, Akif, for presenting today. And thank you, everybody, for attending. And we will see you back at the next webinar next month.
Thank you.
Thank you. Have a good afternoon.